Assalamu Alaykum Rahmatullahi Barakatuhu. Bismillah walhamdulillah wa salatu wa salam ala Rasulillah sallallahu alayhi wa sallam wa man wala. Rabbi shrah li sadri wa yassir li wahlul uqdatan min qawli. Inshallah ta'ala, shortly we'll be hitting BIOA02, Article 39 of week 2. Barakallahu. Just a kind of reminder, perhaps in the midst of completing the lab exam, just bear in mind for those that they pass on the board, because maybe the even if it's big, so if you go into it's deep, you can actually place your small belongings inside from your backpack into the cabinet itself, so that way it's not a tripping hazard. You only run the risk of having to be close to your phone. So just want to whoever completed with their last quiz and able to take stock about their topic. Just before we begin. And reminder for those of you that are starting or working on their lab quiz to bear in mind that it's independent. You can use your lab manual, and if you need it, you can also use a calculator. Just to bear in mind it's 10 minutes, so it's timed, and so bear in mind that you're able to understand the concepts in advance to help prepare you for today. Just want to ask for those of you that just came in, if I could grab your names. Last name ideally. And community.
You couldn't find it was funny. On my way here, I left my bag full of my manual and my copies on transfer. I never do that. I just is it okay if I go to the bookstore and grab it in the lab coat?
That's
During
possible.
my own time.
Yeah.
Okay.
If
So
that's
when
okay.
things I know.
No, the thing is,
Sorry.
if you can, I'm gonna find out if we have an exit here, if you can.
Okay.
No. Okay, in terms of the lab manual, if you use mine.
I have the online version.
Okay. Do you have anything to write on, like paper?
Yeah.
Yes. Okay, so because you'll probably be making some notes, which are
I
difficult
might
to
do them
do.
on my iPad. So.
So you have your iPad with you.
Yeah.
What did you do on time?
AO1 manual, which was in that, because the AO2 is still on back order last I checked. Like I haven't been able to get it in the bookstore. And my coat and my glasses.
Okay. You're being honest.
I drove to the station and then I took a train.
Oh, Why you left it in like go back to the Western?
Yeah,
Your belongings?
just the things. I brought my bag because it was a separate bag.
No, I'm gonna sit down and just see if you can get yourself ready.
Okay.
If you need a computer as well, we have one.
I have my stuff. Just it was just the other one.
Okay.
Okay.
If you turn around, please, if you want to buy, you can.
Yes.
But,
Will you check if there's actually alcohol? Otherwise, I'll go to the bookstore.
I can check.
Okay.
I'm just worried they may not. If you can,
Can I go in?
yeah.
Okay.
Okay.
I will.
Thank you. No problem. Thank you. So for those of you that are still completing the timed lab quiz, you can complete it in the next 4 minutes roughly. We have that amount of time left for today's practical. And of course, so if needed, if you haven't yet completed it post 10:20, then we'll be able to move onwards. But I'm just gonna check if there's anyone else. Anyone else is missing? Is Bruce Santos here? Simone Mack? Owen Parsons? Just want to kindly request from the event, please pick up your backpack. Actually, please by the cabinet, as well as the first one up. Whoever wants to, highly request that you take it all please. If your bag is too bulky, just remove some smaller objects from the bin, and then like, yes. Inside the cabinets.
Okay.
If there's smaller items, like if it's too big and like big to fit all together, you can take out some pieces or some things within and just fit them inside the cabinet with your backpacks. Yeah. If it's too big, because sometimes that helps to lessen the load and burden of your own backpack, and it could fit in more longer essentially with the other smaller objects inside or out, like pretty much inside the cabinet, but now just outside of your bag. If whichever belongings you have that you're comfortable with doing that with, If it's too big already, then you can essentially select accordingly. Were you able to also do the lab quiz or? Yep, perfect.
Question. When you say cabinet, do you mean like those to the left?
The ones where the so you see where like to the right, like
Yeah.
that's where
I don't know why, but
and for any of you that came in a bit afterwards, so hello everyone. Alaykum. Barakatuhu. So I want to say, hopefully everyone can hear me, with or without the mic. Now, a few important things in case you came from another practical or you didn't come last week. My name is Mohammed Saeed, graduate here from CSB, Cell and Systems Biology. And alhamdulillah, happy to be here and teach you guys now week 2 of BIOA02. So if there's any questions, of course, I just put in my name and then at utoronto.ca. So if you have a question for me in particular, just feel free to reach out. Of course, though, you also have the BioLife email that could also be very beneficial. And as we'll see from upcoming slides, the different resources they provide for your FLRs, your formal lab reports, the written assignments, and even just being able to successfully proceed throughout today and other week practical. So any questions on that regard? Especially like for those that just came in, any questions? No questions, okay. So before I move forward, few reminders of safety. And actually, hopefully everyone has by now completed the lab quiz. If you haven't, you could still proceed if there's time left for you. But in the meantime, I just want to be able to go through today's practical with ease. Inshallah, when we finish an hour early. So that will be pretty exciting. Maybe 50 minutes. But hopefully, nonetheless, let's see what we can do. And so my goal is for you all today is to really understand from lab 2 of this week the importance of being able to observe and take readings of different flowers, different plants and their morphologies. We're going to talk about them in some time, but if anyone else, if anyone else is just came in, just don't forget to please keep your belongings inside the cabinets and on the coat racks as well. um, let's see. So, yeah, safety is something I wanted to talk about. We're dealing with sharp objects this week, objects like microtomes. Think of it like those slicing machines for meat, for example. So you just have to bear in mind that we have to be very cognizant of how we're dealing with these sharp objects and that we don't cut ourselves. But in terms of those that aren't aware of what where the objects are in our practical. They're mainly here in the middle bench, and we have some other objects, pretty much the plant containers from 2 weeks ago, shown at the very back. And we'll be making use of them today for exercise 3. So I'm just going to go quickly through. Hopefully everyone is counted off. If Ace Cantos, Owen Parsons, or Simone Mack is also here, they could also raise their hand accordingly. Okay. Oh, and in terms of safety as well, make sure that in general you're wearing your lab safety goggles or glasses. You have your buttoned up lab coat. You are also having your hair, if applicable, tied to the back. So just bear in mind that I obviously want you to be all safe, and for future practicals and courses, you also want to maintain your safety and integrity as well. I'm gonna go straight ahead. So for those that don't know, FLR, formal lab report, is coming up soon, right? And so when it comes to, and actually before I even mention this, if you're ever using your phones here in the practical, just let me know first. Very important, especially maintaining integrity once again, that we don't get distracted. And I also don't want to dock marks for any of you, but you just have to maintain that level of integrity for yourselves. So our details for the formal lab report will be posted on January 31st. However, from what you already see from your lab manual, you could likely prep well in advance. So my advice for all of you is to see how much you can prep in advance before even all the information is delivered on the 31st. So that way, inshallah, you'll ace this FLR. And I'd be more than happy giving hundreds, lots of hundreds if I can. So just bear in mind, do your best, and leave the best with the rest. And so submit your FLR through Quirkus, yes, by 12 p.m. So bear in mind, it's 12 p.m., not 11:59, like it's noon, not midnight on Monday, March 3rd. So just bear that in mind, the different deadlines. And this FLR is worth 5% of your grade. So just also bear that in mind. And it revolves around what we've done from labs 1 to 2. So this week, included. So just bear in mind the methods, the intro, and we'll talk about this in due time. But if there's questions, also feel free to raise your hand. So we have the intro to the scientific method. Let's see timing. Okay. I don't want to waste too much time. Okay. So I'll try to do this quick. To start off, we have the scientific method, you likely saw it in your lab manual. And I think what I really wanted to focus on to give you guys context is the concept of really, once again, going from communication to exploration and thereafter investigation. I touched upon investigation in my I-5 approach. For those that aren't aware, I've incorporated like a multidisciplinary, interdisciplinary approach that's quite unique. And being able to, after 7 years in research and academia, provide a holistic mode of you being able to excel in different ways / modalities. So, and if it's a bit too complex for lexicon or vocab, just let me know. But the idea is that, the scientific method has its pros, but for us, I want you to really focus in reality how you could improve and augment your scientific inquiry, and more importantly, your performance within and beyond this lab, this practical course. So when we talk about a controlled experiment, what does that include? Imagine the experiments you're doing today. We'll likely be doing, actually looking back at our previous week's results from the growth of the different treatment groups, right, from control to treatment 1 and 2. So think about, the controlled experiment that we did. What species did we use? Did we use multiple species? Did we use Aropis thaliana, or did we use the winter wheat? Did we use, like different organisms as well? what was our sample size, right? How many, like we used 10 mils, right, for winter wheat seeds. So bear that in mind. And then the independent variable that you manipulated and actually are being compared between blocks, right? So we had controls at the very back row for me, front of the class, and then the middle row was treatment one, and then treatment two was more closer to me. But the point is that we did also reps to see. So like replicate one, which was like rep A, and then rep B, essentially on both sides. So that way we have 6 different groups to analyze results from. And then you have the dependent variable of being able to measure what we're going to do today, and that's shoot length, i.e. the length of the leaves themselves. And I'll show it hopefully near the end if anyone reminds me pretty much to lift up the projector board. But the idea is that we have 2 main variables that we're focusing on, shoot length and shoot biomass. So bear that in mind, and they're different variables of millimeters and grams respectively. And then you have controlling other variables, right? Like we controlled for lighting, as we did actually on that side. We also controlled assuming for humidity and temperature. So these are conditions you want to be well versed in preparing yourself for future experiments, future practicals, and future courses. And so that's something I want you to all keep in mind. For a research hypothesis, generally speaking, you want to, as shown in the lab manual, be specific to the conditions I mentioned earlier and see how they could actually be tested, right? So being able to define certain or to hypothesize certain results based on what you've observed of reality or even of theory. And then falsifiable, such that if it could be true, if it could be proven true or false, as done and as we'll be showing shortly. An example of this for previous and actually one of your upcoming labs, I think for next for 2 weeks from now, lab 3, it will be like this is an example of research hypothesis. Temperature will have an effect on heart rate measured in number of beats per minute of water fleas, Dafiana, and then the species, subspecies or species category. Now, bear in mind that temperature is 1 unit, right? It's your dependent variable. Sorry, your independent variable. And then, because that's what you're able to control. And then heart rate is what your dependent variable revolves around. And that's where the number of beats per minute is involved as your unit. And then your water flea is your species. Any questions regarding the research hypothesis? Is that self-explanatory or are there questions regarding? And don't feel shy. Of course, I'm more than happy to answer questions. So now, and this is often a question that is often asked, and I also saw this from as like previous experience, starting the prediction or stating the prediction is something that's different from a hypothesis, right? Because your hypothesis is what you're going after or what your quest is, right? What you often are asking about, like a research question. But you're actually providing some more context, right? Regarding, okay, this is what I aim to study, like how temperature will have an effect on heart rate, and then using these units in a specific organism. Now, when it comes to, and it's also shown similarly here, but you're doing like an if-then statement, meaning that if your hypothesis is true, then you would expect X, Y, Z, shown here in blue. And so that's a simple way of understanding it. You have experimental groups that you're focusing on over the control. And then this is actually a great way of actually preparing yourself for the FLR. So think about it like this. What is the effect of drought on growth of winter wheat, right? And obviously, as we talk about things down the road, you can even make it more specific with the different variables we'll focus on today. Any questions? No? I'll go quickly ahead. If you need me to go back, don't worry. I just want to also make sure that we're able to complete as much as possible for the practical. So we did these treatments 2 weeks ago, right? And so what we want to bear in mind is this is actually part of your methods in reality, right? So Think about it like this. All of those, once upon a time, those seedlings, I'm trying to see if we have here. Okay, here's a good example. One is the plants or the leaves that you see here. Generally speaking, you just see, right? You um, just imagine essentially this was a few 2 weeks ago, and then you did experiments to actually treat different conditions, right? So like if the control once again treatment one, i.e. Semi-drought, drought being treated for every other day, and then treatment two for every 2 days, right? And so last simulation is something that's being doing here, but you actually aren't going to be seeing the results down there. What we're going to do for that component of today is really just separating the different pieces of the container to actually be able to measure shoot length and biomass. But I think I'm going a bit ahead, so I'll just slow down a bit. Pretty much, once again, um, control is watered every day, drought treatment one every other day, and then treatment two every third day. And keep note of the different controlled variables, right? These ones are kept identical between containers. And that's something that you want to bear in mind, generally speaking, because it provides you evidence as to what you would actually provide in your lab report, but also be able to understand how it relates from theory to practice. Any questions? Okay. Also bear in mind you're not using phones. I can keep an eye, but I want to make sure you also are paying attention. So let's get straight ahead. Answering. So this is actually very good. I think it will be in fact very important for you for your FLR to think about these questions. Think about the ones that we just talked about from the previous 3-4 slides ago, right? The study of the species identity, your sample size, the independent, dependent variables, the controls as well, and how they would otherwise be controlled, and the limiting factors, right? Limiting factors of what you would expect to be controlled. Now, this is where the exercise for today is. And before I move forward, any questions? Any questions? No? Okay. Yes. Yes. I don't mind, but just bear in mind that, whenever you're using your phones, you always tell me. And reason being is, let's say someone else comes in, a lab technician or one of the profs, right? If they say, oh, why are you having your phone off? You could tell them, oh, the TA allowed me to take a photo. But if you're using it to go on TikTok or do other things, then whose fault would that be, right? So you just have to bear in mind that you're not like getting yourself distracted and also not getting others delayed in the group exercise, because many of our exercises are done by groups. So it's very important that we're focusing and paying attention and not letting anyone else carry your own burden. So just bear that in mind, everyone. I want you all to succeed, but you just have to do your part. Thank you so much. Any questions, though, regarding that or anything else? No? Okay. Perfect. So, and once again, if you have to use the washroom or if you need to stand up, wash your hands, just let me know. No pressure at all. Now, exercise 3. So if you go to your lab manual, we're going ahead from exercise 1, your pre-lab quiz, to exercise 3. And this is where we're going to actually go back to the past 2 weeks ago and focusing on now taking your results for your formal lab report. So just bear in mind, you're being very diligent, very vigilant as well, that you're preparing yourself for your formal lab report to do in a few weeks, right? Think of it like that. So try to think even in terms of methods right now as you explore and actually uncover the results. What limitations are at play? What things could have been done differently? Are there variants that you would have expected or would have not expected, and why? So think of these things and the implications of them, as I mentioned 2 weeks ago, as a common theme for excellence. Now, any questions? No questions? Okay. So what you're going to do is within the same blocks. Yes. Yes. So we're going to talk about it soon, but it's I want to be able to cover everything from the slides just to make it easier for everyone in case you need to go back or have any major questions or concerns. But generally speaking, we're going to be recording the results. You will also look at qualitatively before quantitative measurements, how your plants look. Like, are there any like signs of necrosis, like dying leaves, or if there's spots, or if they're turning yellow in color, things like this that you could observe and make note of in your lab manual. And you could provide different sections in your lab manual to make note of these differences. Now, I'll show you this now, just with one example. Don't worry about oh, actually, this is good. So I'll do it now. I'm not going to say because we're going to be for me, but this will be for you for us. Yeah. You did one team. So you can start making observations, and I'm going to show you pretty much what you need to do. Um, and it's so note of the different observations you see qualitatively. So look at your results, observe your plants qualitatively, and make note of them now for the next few minutes or even few seconds, really. You can start jotting down the different points. There's even questions about them in the lab manual. So just bear in mind that you answer them appropriately. So qualitatively, you want to observe your flowers or your leaves and see what questions you can answer. They're shown in page 41. So if you have time to actually look, you could be able to observe those different changes. So make note of them. Also, bear in mind that when you're doing your FLR, make sure that you refer to both the general name, the common name, which we know is winter wheat, but also in brackets and in italics, the scientific name. Full name, when you first mention it, should come Acivitum, and that's the name or Acivitum in Latin. Now, bear in mind that if you're writing in the formal lab report, you write the full name first. The first time you write it down, and then for future references, you write it down in short form. T A civic. So that's just the point I want to refer to now for you guys for later in preparation for future practicals. Now, let's see. So this is what you're going to do as groups, groups of 4. You're going to take out, you have 2 options. You could either take out, and you can also take photos if you want. You could take photos of the plants before you essentially dissect them. Because you're going to be doing 2, you have 2 options to actually take out the different seedlings to measure their biomass and length. And now is actually a good time to okay. Now, Okay, I'll show you guys what I'm referring to. It's actually this side right here. So the idea is that you're gonna be, you have really two options. You can cut. Is it also show it here? No. So I'll just, let's see. Um, the idea is that you are really cutting down your container into, you'll take out literally the plants, right? So if you don't mind, I'll just do it. I can imagine you had it here, just so everyone can visualize this. You have your container, you lift up your seedlings all the way up, you take them out of the container, and you're literally going to cut them into three parts. From the three parts, you select randomly seven from one side, six from the middle, and seven on the other side. And so from that is what you're going to actually be measuring the different shoot lengths, how long the different leaves are, and or the shoots actually. And then you're going to be taking them out individually to measure the biomass. But we'll talk about that actually. I may already show here. Yeah. So once, and I want to make this as straightforward as possible. So definitely if you have questions, let me know. But the idea is that, let's say, gonna lift this up. So you're gonna be taking them out, right? So sorry, let me clarify. So essentially, you're gonna take out, let's say, essentially from, if you see this, you take it out right now. The idea is that you're going to split them up, right? You're going to choose seven from one side, six in the middle, seven from another. And what you're really arguing is taking out seven leaves. So if they're connected to like multiple seedlings, that's okay. Just take out seven that you're going to be observing for their length, and maintain them on their within the seed itself. So a little bit more like don't know if we have samples here, but the idea is that it looks like that diagram right now. So you're gonna have the length, the shoot being measured while still intact with the seed. So that way you can actually straighten out the shoot and measure accordingly the longest diameter, or even like the longest length, the length of your shoot. Then afterwards you can snip it out, right? So that's going to be done for all seven, even if they're connected to one seed or multiple seeds. And then you're going to actually measure them on the mass balances that you have here. You're going to, when you're at that step, let me know, but the important point here is that you zero your balance before beginning. Make sure before you begin that when you press zero or here, you have different mass balances around here, but each group of four should already have, I believe, an option to here, right? And so the idea is that they may be at times sensitive, but jot down the first observations, first measurement that you see, and that way we could go forward with collecting these data points for your FLR and what will be uploading onto Excel. So that's a bit about what we're doing now. Any questions regarding that? No? Are we ready to start observing and quantitatively measuring the different shoots? Yeah? Okay. All the best? Yes. Any questions? Yeah? Okay. Okay. I'll go with me. Okay. Yes. So to clarify for everyone, once your group is done recording the data points, and it'll be all 20 for the shoot measurements and for the biomass. But once you're done, all 20 from your group, you just let me know. You'll be actually uploading them onto the computer, and then we'll all have them collected to be
uploaded and shared with you on Quarkus. Yes. Sorry, ABC. And sir, your question was it a single one or no? Okay. So, so that's once we're done before I can open it, we'll actually all of them get to I'll have a look from the Excel sheet. Yeah, so we actually have 40 points here, which is why it will be so for your reps, so don't worry that it goes up to 20. Technically on the Excel sheet that we'll be publishing, we'll have all 40 for both reps, i.e. Control A and then Control B included onto Excel sheet, and likewise for treatment 1 and 2. So I would just say for your own observations, record what you see, and then qualitatively, we're going to talk about what observations you see to answer the questions. Because those are questions that are asked also in your lab. Is that okay? Any other questions? Any questions? Don't worry, I don't mind answering more. But also if you're good, then that's also great. Everything okay? Yeah. So if you need to cut your essentially assays, the plants, what you can do is use the scissors shown across. You can have different items to use actually. You can use the scissors as well to cut through the fruit pieces to make the three thirds essentially. You can either pluck them out, which is also really good, or you can cut them down. Like pick 7 for the one side, 6 in the middle, and 7 on the other side. So that makes taking out the seeds, bear in mind that you're plucking out 20 different seedlings, such that you're only measuring one of their length. Also, the other option is that you're taking out only 20 shoots, right? And they can be connected to different, like more than one seed. That's also okay. But just bear in mind that you're doing it consistently. Also, just want to see like that may make it easier for many of you, because I noticed some of you are saying that there's variability. So if you wish, you could just take out 20 seedlings. 7 on one side, 6 in another, in the middle, 7 on the one. So, also to clarify, you're measuring the seedlings individually, or the seeds, the shoots. The shoots of your seedlings individually, just bear that in mind. So once you're done, you take out the seed itself, and then you're actually going to be measuring the seed, the shoots individually. I know it's a task, but do what you can. And we could probably be complete soon, but let me know exactly. Also, don't forget millimeters is the units that you're using, not centimeters. And once you're done, or whenever your group is done, we could be able to count out the different values. If as well you have any allergies to grass or TBO, which is a stain that we'll be using, you can also use gloves for today's practical, if you wish. But if your mass turns to zero for whatever reason, record the first observation, and let me know if you need help on tracking that down. You can also wear gloves for the practical. So how are we doing so far in terms of timing? Good? So if there's any questions, let me know. But generally speaking, I hope it is a bit self-explanatory what you're doing. I know it may take a bit of time, but if you have the potential of having members of your group split up, that may make it easier for accounting for size versus biomass. Just keep in mind the same plan or shoots for biomass and length. But I would just say
That's all right.
How are we doing so far, everyone? Good. How many of you have completed 20 measurements for length? 3 more left. Okay. Perfect. Oh, combined? So you've done 37. Perfect. Okay, great job. Control treatment A. How many have you done in terms of measurements? Okay. Okay, how many do you have left? Okay, okay. How many more from control treatment B? Wasit, how many units do you have left? Values. 7 left. Middle group here, how many do you have? 12. Okay, how many left? Side. You just started for the weight, but you're done the units for left, right? Perfect. Okay. So I'll give you guys about 5 more minutes. Hopefully that should be okay. And then afterwards we'll pretty much upload them. If your group is done accounting for units, for length and biomass, come up to the front and you can upload them or insert them into the Excel sheet. Thank you. Any questions, concerns? Okay, you're ready to upload them? Perfect. So before doing so, I'm just making some edits. So how do they look so far? Did you calculate the mean or? You don't have to do it, yeah? I'm just curious. What average values do you see?
If we talk about the bottom,
Yeah.
I would say the 0.7 was the 0.07 was the highest. Overall, if we calculate the 0.04, it would be
And this is grams, right?
yeah,
Yeah.
0.04.
Okay.
And if we talk about the length, I think 80 is fair, right? 80, 70, between 70 and 80 would be the average.
Okay. Now you were 3 minute one. Perfect.
Should I
Okay.
just keep on saying?
You can actually upload them. I'll make it even easier for you guys. I just want to see.
Can we take pictures and upload?
I will share them to you all the quantitative data, so you don't have to worry about taking photos of this yet. But I just want you to be able to upload them, and essentially I put replicate just to make it easier as well. Because your group, let's say 1A, you could do essentially the treatments all the way to 1 to 20 essentially for both sides. This would be for shoe length, and this would be for biomass.
So we're just looking at qualitative, like 2 mile qualitative observations looking at other people.
We're gonna actually talk about them.
Oh,
Don't worry about that. Just so everyone's clear, if you can have a look at the front, everyone. Once your group is done both measuring 20 units of shoot biomass and shoot length in millimeters and in grams, you can upload them into this Excel sheet. You have shoot length on the left side and biomass on the right. Make sure you're using the right units again. And for replicates, my left side or the right side of the practical room is going to be replicate A, meaning you're going to go up to from 1 to 20 in the Excel sheet for number of seedlings for both sides. And then for my right side or the left side of the room, it's going to be pretty much from samples 21 to 40 for both sides. So just bear in mind that you put the right ones and they're normally left blank purposely for you. And if I have questions, I'll likely come back to your group. But we'll come together to talk about the qualitative observations afterwards. First off, if you're ready, you can come in and upload your data.
All right. Yeah, right.
So I can just want to see if you have access. Okay, I could just upload them onto yours.
Thank you.
No problem.
Read it to you.
Okay, yeah, read it. Oh, okay. 73?
Yeah.
Keep on saying.
75,
25,
40,
40,
Later, to me, it's a bit more time.
92,
For today's practical,
54,
the leader class,
67,
84,
84, 55, 63, 92, 73,
70,
70,
104,
104, 64, 90, 95,
90,
90, 127, 64. Done? You bet.
the mass,
Oh, not yet. Yeah. Let's go. 0.06.
0.0. Zero point zero three. 0.03. 0.05. 0.03. 0.02. 0.03. 0.03. 0.02. 0.03. 0.04. 0.04. 0.05.
actually, group treatment 1A, that's the come out.
All right, done.
Perfect.
Okay.
No problem.
So you can upload the links here. And then biomass here, just make sure you have the right units as well. You can look. If you wish, someone could share them, like one of you could see them. Or one of you could
Too much pressure. We have to start there.
Yeah.
Someone tell me.
We do this,
Wait, regular watering control is yeah, this is
right? Yeah.
yeah.
Okay,
Wait, did we do it right?
Because you didn't do the treatments, we had the control.
Wait, double check.
The name title. Oh. Control. Okay, yeah.
So
108.
wait. 108. Just 108? Millimeter.
But that's
Okay.
and 91.
Okay.
So let me. 74.
Wait.
So what is 81. 8.
Was that small? Wait, sorry, what? 53?
Yeah. And 89. And 78. And 58. And 73. 63. 75. 74.
Was that all right?
If you also want to take photos of yourself, like doing these observations, that's also.
Oh, it's fine. I thought I added the row. Is that it?
You got all the implications of your result. This will help you prepare the date of your FLR.
No? No?
Yeah.
Point 0.05. 0.05? Yeah, 0.0. And then 0.04. 0.0. 0.06. 0.0. 0.05. 0.04. Again? Wait, oops. Wait, sorry, I forgot the desk. Yeah, we done?
Oh, did you become a I can help you. I have a feeling, so the number lock is on, so now it should help. Yeah, I think it, yeah. If you keep going up, actually, you keep moving it all the way up, you should be able to reach it. How many were you planning? Like, where did you start off? By the way, to confirm, these are two separate leaves, right? They're not the same. Okay, okay.
Will I use this? I can use this.
You can. So everyone, I just want to make sure you're making full use of the lab. If your group is done or is waiting to upload the results, this is your time to start preparing for exercise 2. This is more of a heavy lab for the final exercise. So bear in mind that you're paying attention that way, especially when you're in. Because I doubt we don't actually have that zero. So I would say the more safe.
3.01? Okay, because it was just very, it was like, we weren't sure if it was between 0.00 or 0.01. We weren't sure, so that's what it
Oh, but how do you
No, because we weren't sure if we should add the zero, like,
I would honestly go 0.01 because otherwise, yeah, I would go with 1, given that.
it was only 2 things.
Okay. Um, you can keep it as that. Then. Yeah. If I see there to be concern, I'll just record it itself. So don't worry. No problem.
See,
Yeah. Oh, yeah. That's
Okay.
yeah, but like it's boring.
Oh, yes. Oh. See something. You're done? You're done. No, just have. You want to use the rest?
Okay, I don't know.
They need to go to this class. This page.
Yes, 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 yes. Yes. I mentioned. I'll make mention of it. Thank you. So everyone, I just want to make sure you're all paying attention. At least for the remainder of today's practical, you will need to use gloves, especially those that have allergies, but particularly for the use of the stain TBO. So I don't want anyone getting any hazardous material on themselves. So start to prep up with some gloves, especially on the left-hand side. I do want to say as well that you could start sharing with one another qualitative data, so what your flowers or your shoots look like. We'll talk about them briefly, but I do want to make sure that we have time to complete the rest of today's lab involving exercise 2 right here. So if your group hasn't uploaded the data as of yet, this is the time to do so. We have one gentleman here, Jaden, got your name, Mahima, right? Okay, and then one more group is remaining. So okay, so the 3 of you can stay here, keep uploading them as quick as you can, and you can validate them as well. In the meantime, I just want everyone to start getting ready for exercise 2. And hopefully, this would be easy. You have to be very careful. I want to say the following. You have celery, right? You have some stock. You're going to be sliced through that. Reasonably. Anyone who wants to see the demo now, it would be highly recommended to come here into the middle bench. So I'm just showing the setup. Once you guys are ready, you guys can come. I want at least there to be 2 members per group here. Yeah, so that way you can validate things in case one person has questions. Here can clarify. Everyone's looking around. And those that are continuing to upload the data, the way you can also have a button. The point is the following. We're going to be taking very thin slices that are essentially this big. So where I have that cut right now. This is pretty much preparing you for a microbiology class because you have slides and you essentially slicing up different organisms or you just visualizing specimens through. But the idea is the following: connected with integrating back plant biology. You're going to be starting off because the microtome grass you can do these big slices. You're going to essentially have to
fit them inside this block. So we take it out. And the idea is that you initially, you just pretty much are, think of it like a car to an extent. You have what you're uploading or where you're starting off with, like a screw at the very back. You're going to be moving it a little bit down, not all the way, but such that you're able to fit this inside. Now, clearly this is too big, right? So what we're going to do is actually cut it off a little bit of its length. I'm just going to leave this here for the time being. So we're going to cut it likely about this much, so that way the remaining can fit in. And if you look at your lab manual, this is pretty much size or the amount of what you're going to be using for your celery section. Any questions regarding this? Like if you look at the thin sections, page 38. So if you want to see essentially what it would look like, is pretty much what you have here. And now hopefully, oh, and you can also cut size because this is quite too big. In fact, I know it looks like it's a good amount, but for the purposes of this exercise, it's soft right around here. And just bear in mind, you want to have a good amount popping out. See how it should focus actually larger. I'm going to cut about maybe 3/4 of that amount. Always make sure you wear gloves, even when you're dealing with the razor here or the razors. I know it's a bit dense or a bit hard to navigate at first, but you just need to make sure that you're safe. You're not touching it too close to your hands. And now you have it as thin as you can. Now, I'm doing this for one group right now, but the idea is the same for everyone else. This could be a thick slice. I realize only one side is the so I could do it twice, but the thing is, it's in, right? So you can see that it's in frame now. And this is where you're slicing. Actually, very sharp. Be careful. Obviously, here is where you're not going to put your finger, right? Because actually, slice. You don't want to touch your body. So, I think this is going to be a thick section. As you slice it down, do this again. Most of you are on this side, so how about if you want, you can just come down this way. We can just do it one time. Because I also want to make sure everyone's able to see this and we can maximize efficiency. Yeah. Now let's see. I'm just doing this for training purposes. I don't want you to actually put your finger here. But I'm just wanting to test, hopefully, that this is stable. For previous occurrences, sometimes isn't. And this is quite thick for my sample. What I could do is, what I'm doing right now is like tightening it up, so that way it's like a clamp. And the pieces that are on this side versus this side, which is going to press it down, I'm going to try to keep it firm in place. Just keep it like firm, and hopefully it's quite big. This is actually a thin piece, and once you take out, once the piece comes out, what you're going to do is the following. Normally, I think it's on your benches. You have 2 petri dishes. Don't see them there, but normally you have 2 petri dishes. One would be for your stain, and one would just be with filled water. Another member of your group could already prepare for this. But the idea is that because you're generally a group of 3 or 4, the person that sliced it could Then after, or with the speaker up here, lift it up with a paintbrush. That way they don't run the risk of actually cutting themselves. I know it's hard to see here, but imagine hypothetically, this is your piece. The pieces are going to be better because generally the first few are harder to actually do. And they're often not straight, so that's why you have them small and sometimes wrinkled. But the point is that once this occurs, we'll do like 5 good sections perhaps. And I want you to actually just test it out yourselves. And then you'll be actually staying them for about 30 seconds of TVO. I don't see TVO here. It's on your benches. And so after 30 seconds, you just simply transfer it with the forceps or with the paintbrush is okay. So you can just thereafter upload it, transfer it into your dish with filled water. Any questions regarding it? Is it clear or do you want me to go through what's
So can we put the cucumber or other lamb slice into the video?
Yes. It's gonna come out. This one isn't coming out because it's too big. And it's good for practice, but let's see. Like this is ideally the smaller, the thin side of what I have here. I'm just going to give you guys a thin example of what it would look like following that slice, because the slices are quite similar. Though that one's sharper, that one.
Yeah. I'll do one.
But this is a good example. This is very good. So, like hypothetically, once you're actually slicing it through the microtome, this is where you're going to actually see, like what I have here, and you're going to see it come out using your paintbrush. You can lift it up or with forceps. I just worry that the forceps are delicate, so try it. It's too hard to do with one paintbrush. Try another, and you can gently lift it up. Okay, well, the other thing is this surface is different from the microtome, so it'll be more easier. But the point is you lift it up, place it into a petri dish, and thereafter you add in a few drops of TBO just around, like it should be across the entirety of your cross section. And then after 30 seconds, at max 60 seconds, so go with like 44 seconds, and then you just place it into the other petri dish with water. So that way it can be diluted from the stain, and that way once you look at the cross section over the compound light microscope, you'll be able to see amazing details of the different tissue plates found in stem. So that's your investigation exercise 2. Any final questions regarding No, actually, in fact, each block will have its own cutter or microtome. So you guys will have, there are 6 slots. I'm dealing with one of them right now. But essentially, we'll even probably make use of this one. In fact, there was one other thing. Yes, make sure that when you're using the petri dishes, you've already loaded essentially water into one of them, so that way it'll make the transfer easier once you add TBO across the surface of your celery stalk. Any other questions? Very important that you're all safe, by the way, when you're held.
Take 5 cuts.
Yeah, 5 cuts. Okay. So you're going to do 5 cuts. You have 4 people per group, right? So you can do more than one. If you need help, also let me know. I want to make sure no one's actually playing around because you don't want to get cut. And just be safe. Follow the steps that you see from your lab manual and plus what I've done. Or essentially, if you have questions, let me know. I could walk them more through with you. Just want to confirm though, everyone has uploaded their data, or are we almost done? I think that's the last group. So in the meantime, let's continue with exercise 2. We'll talk about the qualitative data afterwards for exercise 3. That's relatively simple, and then we can end off today's week
talking a little bit more about the formal lab report. Um, any final questions regarding this? No questions. Then there are other steps for exercise too. Once you look at your light microscope, you can record the data. But in the meantime, and then you'll actually calculate the size itself for different regions. You just bear that in mind, everyone. Once we reach there, I'll let you know. But try to finish this in about 20 minutes. I think, I don't know, 11:52. Okay, so I would say if you can even just do it shorter than that, that would even be better. But let's say 12:05. Try to be done by 12:05. This step, then we'll try to complete the rest of the process. Yeah. So we have to initially cut it quite large. So you have to cut it from one angle, like the length, to fit it in. But also it's also quite deep, like it's quite long itself and wide. So when it's wide, you cut on one side, but when it's long, you're actually going to try to cut it just a bit so that way, like once you fit it in, you'll be able to cut the overlap. Exactly. And the slicer would be this essentially. You just have to be very careful. So I'm going to do. But yeah, that's pretty much what you're doing. And did that answer your question? Like any other questions? No. Yes. I'm not sure about other so I think I have to go. Don't cut yourselves. If you need 2 gloves as well, like double gloves, like double gloves yourself, that's also okay. Just fill up the dishes with water. Oh. Okay.
Right here.
Yeah, like it should follow everything, right?
So I just want to show, not like these are the types of slices you want to take actually, if you can. It's they're thin, right, like you can imagine. And if you could do it, you did it with a microphone. Okay, so it's good as is. If you can do it with the microphone, that's preferred. I would highly recommend doing it. It's also a great way to prepare yourselves for future microscopic practice. And even just for learning more about microphones themselves, because other apparatuses use them, like scanning electron microscopes for preparing samples. And this is a great way to so just think about the applications of the tools you're using. They're very beneficial and also applicable for your as well. So keep that in mind. Try to do as much as you can. If you have questions, let me know. I really just don't want you guys to be very good. Let's say sorry. Everyone's paying attention, like focusing essentially. If your group is done, taking up the one to 30 minutes, just feel one, you just really want to think about it.
until you actually mount it on. So if you are done, let's say one slice on the microphone, all you need to do is turn it more because it's gonna push it further. It's like it's. I'm trying to think of another way to describe it, but essentially this is gonna turn it forward, so that way you can slice and you can continue to repeat it from that sense. And you're only doing that, I think. This quickly gives you an amount that's like pretty much 30 degrees. That's 60 degrees. So essentially make like a half screw, and then by that part it should be enough. If you were to add it individually, so you're gonna take your time. And then Both should be okay. If they're too thick, let me know. I'm happy. Careful, everyone dealing with the microphone. You don't cut them. Okay, clear that in mind. 50 is. Pretty much open the surface. Make sure the drop of PPO covers the entire of your celery cross section. Don't let it just be on one side or one half, but the entirety. It should be enough water. Maybe yeah, it's all.
Oh, I know what you mean. He went to the.
It's actually back.
You start reviewing the quality of the wells. Also, everything right now, it should be done almost daily, if not observing your celery stalk cross section. Have at least five, so that way you can also record the observation for the time. I'll be backwards.
Okay, that's enough. Well, the stalk is like. I think it is.
So everyone, I just want to make sure you're all paying attention now. Hopefully, there's a few lessons, hopefully that some of you can also consider for upcoming practicals, but also if you haven't completed the celery staining, what you need to do now. A few things I noticed was essentially the amount of stain. Some of you have asked, how much should I add? Pretty much up to the surface, the surface of your celery stalk. Not all the way up to the top of your petri dish, but just a very small, like maybe 3 drops, 1 to 3 drops at the max. But then when it comes to the timing, it's about 30 seconds at max 60. The question though is that when you're rinsing it off, some of you have not been rinsing it fully, such that the stain is maintained. And unfortunately or sadly, there are the cases where if you haven't used the microtome and it's too thick, the stain would have already been absorbed readily, such that now it's too difficult to observe. But at least from the naked eye, even though that may be the case, have a look. And perhaps it may even be better to visualize under the microscope. So I would say if you can, continue to rinse off the stain. If it doesn't after 1 minute come out as much, or if it's blue as in the residue of remaining liquid, then just dump it off. Add in some extra water, and then continue with the cover slip light microscope observations. Very important that you maintain well sample preparation, so that way your microscopic visualization is easier. Any questions regarding this? No questions? Are you guys able to understand? Everyone heard what I said, or do you need me to repeat myself, or everything okay? Everyone, make sure you're using new fresh water when you're rinsing off your new cross sections, because otherwise you're going to maintain the stain within your celery stalk. Bear that in mind, especially I don't want anyone running the risk that you've done amazing cross sections, but the staining, if it's left too long, may be difficult to observe under the microscope. Just want to confirm everyone's uploaded their data. Is anyone still waiting to upload their group's data?
I mean,
Okay, so I just want to go over some other things. I'm briefly gonna also look at your results. But I want to make sure everyone's also up to pace with what they are required to make a note of. So we've passed this step, right? We're past exercise 3. If you've already stained, who's having problems observing their cross sections? Okay, I'll come to you all shortly, or to the different groups that need help. But let's see. This is what you could see from the TBO stain. And make sure you start focusing. Once you're at the microscope, whoever's still waiting to restain their cross sections, let me know.
And then the next week, they can get even a little better. Put them out there, then everyone found out.
Let's see.
I think this one's over. Yeah, you can see that on the target.
So make sure you clean up your stomach, especially that are left at the waist. You can return them back to that feature which has the problem, celery stalk.
No worries.
So, question. Oh, wait. When you're done with the TBO, please put the waste down in that in that fume hood, pretty much that bin where TBO waste is labeled. Please don't dump the TBO down the drain. Place it back into that beaker. Thank you. Any questions? Yep. So everyone, okay, I wanted to make sure that we pass through hopefully the rest of today's practical with ease. So please make sure, kind reminder once again, you're not using your phones, you're paying attention because there's no need for me to dock marks unless you want to. You want me to. So just bear that in mind. I'm going to go through a few other important points. You please need to pay attention, right? I don't want to be impatient. I want to make sure you guys are all able to focus. And for those that are paying attention, they don't feel distracted or delayed in timing. So let's go straight ahead. Back to what we were talking about earlier about exercise 2. We didn't touch upon this earlier, but the summary of plant organ function, because you have different plants that you'll be focusing on. And there's some of them are on the right-hand side of today's practical within the projector board. So you want to just think about a few things. Based on the lab manual, we have simple tissues and complex tissues at the finest, right? When you're looking at your light microscope, you're looking at different types of plant cell types, right? And different types of tissues. Amongst the different tissues, you're going to be taking, and please pay attention, everyone. If you go to page and this is exercise 2. Page 37, you have different cell types, right? And you need to make reference to drawing 3 to 4 of how they look for the different categories that are shown for celery stalk. And then you're going to be mentioning what their colors are, right? And that refers to, as you would have expected from the previous table, the different types of walls. Are they a primary or secondary cell wall? What's the difference? You'll see that also in your lab manual, but we'll go through it briefly here. As you know, plants have different functions, right? They need light, oxygen, and the nutrients that are involved, water as well as CO2 that relates to their essentially respiratory and nutrient uptake. They also have carbohydrates important for photosynthesis, and vegetative organs, tissues, and cells allow for plants to receive, transport, and maintain these different molecules or biomolecules. So in exercise 2, we've already gone ahead, but I want to make sure that you're able to understand this clearly in prep for your lab exam. So understand that there are different components, once again, of plants, the reproductive organs, leaves, stem, roots, and above the roots are your shoots, the components that we explored for today. And that includes your biomass and length that we focused on. Function of a stem. Does anyone know its importance? What's the importance of a stem for any plant? Yes. Uptake water, yes. Yes. Another one, yes. That's also very similar. So both key components of structural stability and uptake of nutrients from the very, from pretty much the soil, right? There are some, interestingly, there's some organisms that don't grow with nutrients or like with soil. And there are flowers known as hydroponics that grow in unique ways without use of mainstream water. Let's go down. So we already have cut down using the microtome thin slices of celery, right, from their stem. So that's something to keep in mind. And we used polythene blue O stain, or TBO, right, for short. And you're hopefully able to now see from 4 to 10 mag or 10 times magnification what the cellular ultrastructure is of these celery stalks. So you're able to see now as you go down and down into the cell what the different components are within your celery stalk. And they are to be completed in table 2.4. 2.4 is on, let's see. That looks different for what we've done thus far. I believe it may have been updated. Yeah. So it's actually not 2.4, 2.3 is the one that we have for the summary. But as you go down, we talked about the demonstration. I've shown you about doing the slide, right? Preparing for what you're to look right now on. And let me just turn up the screen. So now you can see what I'm seeing. Okay. It's fun. It's a bit of manual work, but it's okay. So look at the right-hand side from where I am. This is what your celery stalk looks like, right? And you have your plant cell wall. I talked about this earlier. You have a primary cell wall, right? Which is important for pretty much the beginning of plant cell development. And they're made of 2 key components, cellulose and pectin. So if you think of mnemonics, primary pectin. That's your main difference from the secondary cell wall, which also has cellulose, but has lignin as another biomolecule. And the difference, as you see, can be shown in the function of it in your lab manual. But what's important here is the difference of color. And that's where you're being able to describe, okay, this is indeed primary and secondary. The difference is that for your primary, once again, following the P, right? Pectin, primary, and interestingly, the color is pinkish purple. So you have P repeated multiple times. So you have primary plant cell wall. It appears following the TBO stain pinkish slash purple. And then for the secondary state, it's bluish green. So just bear that in mind. When you're looking at your microscope, you'll likely see that difference. Even if you stain a bit more, hopefully you should be able to see differences at the edges, which is indicative of your secondary cell wall, and more within the frontiers or more inwards, you'd see your primary stain, which is pinkish purple. Think about the different expectations when you make note of this for your observations and what you would have expected if not present. Now, when it comes to table 2.3, as I talked about earlier, page 37, you have different components now of your primary cell wall, right? Who knows what 4 of them are based on their lab manual? 4 different components of your primary cell wall. Anyone? Have a look. You can even look at your lab manual. I hope it's to an extent good prep for your lab exam. Yes. Parenchyma is 1. Yes. Phloem is yes. Yes. Good job. Xylem is as well. One more. Starts with the S. Yes. Yes. Okay. So bear that in mind. Oh, sorry. C. What's the C? I know it's there, but it is C. Yes, sclerenchyma. Yes. So, and then the secondary is the sclerenchyma, and I believe it's the xylem. Yes. Now, just to clarify, no. So someone did say the xylem earlier. I think it was Wasi. It's epidermis. Epidermis. So bear that in mind. The importance of this is the following, because now that you know the different substructures, you should be able to see based on your figure, which is we talked about as page 39. You should be able to note the differences in the different components, such that now you're able to label it now in your lab manual on that same figure. So keep that in mind. Also in your table 3.4, and hopefully this is like near the very end of today's practical, so just keep up with staying up to date. Stay focused, hopefully. The point is that you need to be able to make note of these different cell types within both cell walls that are found in your celery stalk. One of them is not actually present, so bear that in mind. So keep looking for at least 5, right? And then be able to note what they look like, what color do they show, and their size, which is what we're going to do as the last component for today. Any questions before I move the projector down? Any questions? Any questions? No? Okay. Actually, I want to make sure you guys are all aware of what I'm saying. So please make sure before I end off, you have 3 different tables, right, that you need to go back to. Just make sure you complete them, as well as the last figure for today, which is what the celery stalk looks like right behind me. Draw it out, or actually it's already drawn for you. All you really need to do is put those points or labels and annotations already along. Remember to only 5 components out of 6, so make sure you find out which ones they are and be able to draw them accordingly. That looks like it should be it for things here. And then that's where we talk about the sizes. Any final questions? Yeah. Oh, with your group? Yeah, perfect. Any questions? Any questions? No? I think I should go up then. Okay. Yeah. You guys need help determining cell size. We're going to talk about it soon. But the point is, what we did last week or 2 weeks ago is similar to what you do now. Look at the cell size based on the OUs and just simply multiply that value, that these 2 OUs at 40x,
multiply that by 6, and that's the number of microns, micrometers you have for yourselves. Keep that in mind when you're writing them down for 2.4, and it's more simpler than 2 weeks ago. So hopefully
I don't know. Oh, yeah. Okay.
want to use both microscopes at your disposal, you can. That would be actually a very efficient. I just want to.
I'm gonna talk about.
Great question. So how's everyone doing in terms of observing their celery cross sections? Has everyone been able to identify the 5 different features? Yes? Now, when it comes to cell size, I'm going to talk about this just so that way we can end off table 2.4, the last column for cell size. So if anyone hasn't done it, let me know now so that way I can let you have maybe 2 more minutes just to write down any other observations.
Yeah.
Done once again with PPL waste, you can make you can place it all down here, not into the same PPO, go inside here. Anyone having also problems trying to identify the cells? So everyone, I know it's a bit tough, but hopefully now we should be able to complete today's lab talking about cell size. Pretty much we're near the end, and we have a few other components, but I want to make sure that you're able to complete the lab with success, so please pay attention. Now that we're hopefully near the end, everyone paying attention? By the way, if you have any extra waste, like the plants as well, you can place them back into your containers or actually throw them into the garbage. I'm trying to think anything else. Make sure you place back, return the items like the scissors, the tweezers into the small beakers that you had. When you're done with today's practicals, you shut off your light microscope. Any unused liquids or bottles, you'll return back to their white trays accordingly. So just make sure before today's practical ends, everything else is clean. The question about size and yeah, pretty much size is the following. Remember, 2 weeks ago, we looked at essentially cell size, right? And remember that you are looking at your ocular micrometer, which is the ruler in your left eyepiece. Now imagine if you see, let's say, a pollen grain, right? Remember 2 weeks ago, or in this case, you're looking at a cell type within the celery stalk. Let's say it's 15 OMUs long, under 40x magnification. Then what you're doing is 15 OUs or OMUs multiplied by 6. That's 90. 90 microns or micrometers is your answer. And so you do that for each of the cells that you're looking at. If you're running out of time, then at bare minimum, you should have at least 4 per cell type. So just bear that in mind. I would say 3 to 4 just to make sure that you have enough time. And to help you out, well, there's one cell type I said earlier, right? That's not present in your celery stalk. Think about the different functions I said earlier. In your secondary cell wall, there is only one feature or one cell type. So that should actually give you the hint away, because generally speaking, all plants have both a phloem and xylem. Generally speaking, or some of them, at least the ones that we're dealing with today. So you should know hopefully which one is omitted from your lab manual, and that's what you really don't have to focus too much on, but focus on the other cell types. Try to take those measurements now. You have 2 different microscopes at your disposal. So everyone, I just want to make sure you're paying attention. In the next 3 to 4 minutes, there should be 2 people per group on the different microscopes. Being able to record cell size, even in OUs, and that could be done every minute for 2 people. That would be a max of 2 minutes for one cell type. Now we have 5 cell types. If you haven't started already, and if you have, then you're likely halfway done. Meaning that in total, you should be done in the next 5 to 7 minutes at max. Once you're done that, then pretty much you can look at the qualitative results of the different types of conditions that we looked at today. In about at 12:50, I'm going to be telling you or asking all of you what those observations are. So you guys can actually already interact with one another if you're already complete. But I would just say at 12:50, we're going to finalize those observations, or by that time that should already be done with the cell size, and then cleanup should be done thereafter. If there's any questions, let me know. The final 5 to 10 minutes will focus on the Lab 2 assignment, which is pretty much you making a presentation. And you can see it also in your lab manual, but the details are also shown here. And what you could also report in, are you able to identify the description? Oh, yes. Pretty much make use of these figures or images from the slides or from your lab manual to answer the questions that you see on Quercus for differentiating the different characteristics of plants, from the xylem to the phloem to the parenchyma, sclerenchyma, and other components, primary and secondary wall. If you wish, actually, yeah, let's just see. Yeah. Thanks.
Okay. Okay.
I'm just like working through it.
Any final questions about all sides?
So everyone, just want to repeat in terms of timing, we should be done shortly. But in terms of the cleanup, as raised earlier, plant waste, I don't want to go in the garbage, but maybe if you want, I'm trying to think. I wish there was an organics bin here. But essentially, you can place it in the U line, black bag. Remove labels for plant containers. Plant containers go where the man who wants it is. It says go here, literally. So just go there. Place the empty containers from your disposed, like essentially the experiments that you had today. Place them back there. And then make sure your lab bench is clean and organized and all personal belongings are removed. Once again, you have a presentation to make. I'll talk about that afterwards, but make sure in terms of being able to discriminate the different features, you can have a look back into your lab manual. And if it's easier, the xylem is generally more larger in size than the phloem. So just bear that in mind. It's more centered, whereas at the cortex, you have the phloem. So just bear that in mind, everyone. It makes it easier to understand.
Yeah.
Yeah, you have to draw those things. Yeah. We can't just use the pictures and circle like but the drawing is just like 5 yeah. So that's okay. That's what oh yeah, right. Yeah.
Everyone, I think it's time for us to start cleaning up. So let me just reiterate this once more. Please bring back the Petri dishes that you had earlier. Cover slip. Oh. Box bin, the blue bin on the left hand side there. The plant go inside the box bin, and then you can make sure you remove the paint from your containers. The waste for TBO goes inside here. Don't dump it in the drain. And make sure that you remove the tape from your containers and get rid of the. Any final questions? So just so everyone's clear, we're about to finish up for today. So I need to make sure all my components are set up in 1 minute. Make sure that the rest of your bench is focusing and cleaning up everything else from the group. I need to make sure that if you have other members that are cleaning up, if others are playing around, that's kind of okay. Make sure you're focusing and being able to clean up because it doesn't add any additional screen for not only me, but the other lab techs. Before you end off, if you're taking pictures, you need to make those 3 things. The shape of your cell within table 2.3, the color from the stain, and the sizes that you've determined based on the OU and calibration factor. That's it for today, everyone. I hope you have an amazing rest of your day. And hopefully in 1 month's time, we'll be able to also have some fun. But I just want to make sure you have, once again, 5 cell types. One of them is only from the secondary cell wall, so bear that in mind. And hopefully that should conclude today's practical. Thank you, everyone. Like, I mean, 5 cell types, if you pick them, you're going to just clean them. I'm cleaning up. I'm cleaning up. I can leave a few slides on if you want, like the slide deck. Oh, that's okay. Like, okay, okay, no worries. Just before you leave, everyone, make sure that you've completed or you're in the process of completing not only cleanup, but your observations for what you've done thus far to prepare you for lab number 2 in its assignment. So bear in mind, once again, your assignment is due 24 hours from today, and it's a 3 slide presentation. So bear that in mind, everyone. Once you're done, make sure you wash your hands. You're free to go. And make sure, once again, you would have completed this with a recording of yourself in the slideshow. Thank you so much. Have an amazing day. No problem. Do you want me to keep the projector on? Or? Thank you. It's a lot of content, but it's okay.